Okay, so what we're going to talk about is environmental factors that influence microorganisms. Uh, it's not just going to be about the nutrition that these organisms find within their environment. Environmental factors, things like temperature or pH, affect how well organisms can metabolize things, okay? Because they have an outstanding effect upon the enzymes responsible for those metabolic processes. So, organisms that are very adaptable and are able to actually quickly adapt to changing in their environment are going to be some of the more complex organisms out there. What we mean by this is that they're technically going to have the ability to use multiple types of enzymatic pathways because that is the way that they're going to be able to survive. Now, I'm not going to talk that much about the enzymatic pathways here, but I am going to talk about complexity of organisms. And one thing I want you to realize is that more complex organisms are going to have more genes for metabolic processes and therefore are going to have a larger number of proteins. Okay, so this is something that is genetic. It's inherent to the organism. Either you have it or you don't, or you have the capability of doing it or you do not. Okay? So complex cells that can switch between environments are actually able to do more metabolically. So our first environmental factor we're going to talk about is temperature. Okay. Now, every organism will have a set of cardinal temperatures. This is the range of temperatures for a growth of a given microorganism. Now, this will include a minimum temperature, which would be the lowest temperature that that organism can survive in. A maximum temperature, where these organisms are sort of at their peak temperature. It's the highest temperature that they can acclimate to. Anything higher than this, the organism will not function. And then an optimum temperature where the organism functions best. Okay. Below are some examples. Okay. I refer to these as the file terms. Yes, you need to be able to utilize the file terms. Think of me giving you an example of an organism and a temperature that it is most comfortable in, and you should be able to tell me which type of organism or which category of cardinal temperatures that particular organism falls into. Okay? We can see it here. Okay? This being a minimum and this being a maximum. So minus 20 degrees all the way up to about 15 degrees Celsius. These organisms would like it particularly cold. 0 degrees Celsius is freezing. Okay? We see them doing not so well. Okay? They're growing really slowly down here at 0, doing much better, and then the growth drops off again. Okay? Here at 15. These organisms are known as psychrophiles. Okay? You can see the term here, so we've color-coded. Okay? These organisms are known as psychrophiles. The optimum temperature range for a psychrophile is going to be somewhere slightly above freezing. Now, I don't use the term psychotroph that much. They like it kind of somewhat cold. But we will commonly hear organisms referred to as mesophiles, with meso meaning middle. These organisms prefer mid-range temperatures, maximum being about 50, minimum at about 10, and an optimum somewhere around 37 degrees Celsius. Okay. Lucky for us, that's the temperature we're pretty used to talking about. Okay. The thermophiles will prefer hot temperatures, somewhere between 45 and about 80 degrees Celsius. Thermophiles peak somewhere around 70. Okay. And then there are those that prefer extreme temperatures, so what we call extreme thermophiles. Okay. The extreme thermophiles will live in extremely hot environments, upwards of 120 degrees Celsius. In other words, they're going to like it hotter than boiling. Okay? And you'll find these in like deep sea vents or hydrothermal vents. So if you think about it, when we start talking about organisms that cause disease, which types of organisms are you probably going to find most commonly inside of a human being? If you said mesophile, you would be correct. We're at about 37 degrees Celsius. Their optimum, or the temperature at which they grow best, is going to be in that mesophilic range. Okay. Another environmental factor, the presence of atmospheric gases. Okay. The ones that are most influential are going to be oxygen and carbon dioxide, with oxygen having the most impact upon microbial growth, mainly because that oxygen is extremely useful when it comes to cellular respiration, and organisms that respire aerobically, okay, will actually make more ATP than those that do not. Okay, so it's going to have impact on growth because think about it, building an entirely new cell, you're going to have lots of energy. Oxygen is both an important respiratory gas and a powerful oxidizing agent. Okay. Microorganisms in general fall into one of three categories. Okay. Those that can use oxygen and detoxify it, okay, which we term aerobes. Those that can neither use oxygen nor detoxify it, so they're not going to use it and it is going to be toxic to them, we term them anaerobes. And those that do not use the oxygen but can detoxify it, we term them aerotolerant anaerobes. So they can tolerate the oxygen, but they're never going to use it. Aerotolerant anaerobes. Now, some of you are probably wondering, what do you mean oxygen is toxic? Why would you need to detoxify it? Okay. Well, the answer is coming up. Actually, I'm going to go ahead and move to here, how microbes process oxygen. Okay. And I'm going to back up and get back to those generalized terms in just a minute. But the reality is oxygen is what we consider a free radical. Okay? That as oxygen is metabolized within the cell, what's made from the oxygen, those byproducts of oxygen metabolism, are actually poisonous to our cells. So cells have got to have a way to deal with toxic oxygen byproducts. Okay? Now, I'm going to come back to this. Don't worry if you didn't write anything down. I swear I'm coming back. Okay? So, let's get a little bit more detailed about those categories of organisms based upon their oxygen use. One thing that's nice to think about, and it's easy because you've actually seen this within the lab, okay, is that within liquid culture, so within a broth culture, you will actually see patterns of growth for certain types of organisms. And if you think about it, it's going to make good sense. The aerobes need oxygen. They're going to be up at the top, okay, of your tube because that's where the oxygen is. So you're going to get the most growth there. Okay. We also have what we term microaerophiles, and microaerophiles prefer small amounts of oxygen. You're going to have to train yourself from thinking that aero means air. Reality is that we're talking about oxygen anytime we talk about aero. Okay? So microaerophiles like small amounts of oxygen. Anytime you see that phile term, that's what we mean. All right. Now, they're not going to grow all the way at the top of the sample. Instead, they will be slightly below the surface of whatever growth media they are in. Okay? Mainly because they will utilize the oxygen, but they can't detoxify it very well. Okay? So slow detox means that you don't want to be in too much oxygen at once. Okay? So they're going to be directly in the atmospheric oxygen. Okay? They're also what we term facultative anaerobes. Now, this is a new term, and everybody should learn to use it. It will definitely show up on the test. Okay? Facultative. Now, I encourage people to, when you see this word facultative, to mentally switch it in your head to can survive. Okay? Because we're going to use it in front of another word, and what we're saying is that they can survive this condition. Okay? Now, that doesn't mean they don't survive in the other condition. It means that these are versatile organisms. So when we talk about a facultative anaerobe, what we're saying is that this organism can survive in an anaerobic environment. Does it want to? Maybe not. Okay? But chances are facultative organisms survive in the opposite environment. Okay? better than they would in that facultative environment. So facultative anaerobes are great in aerobic environments, but if something should happen, okay, and let's say you're growing them in a test tube that has a screw tight cap, okay, and instead of leaving the cap open so that the oxygen can get into the test tube, you happen to close the cap tightly and leave them over the weekend, facultative anaerobes are not going to die. They can switch from aerobic metabolism over to anaerobic metabolism. They actually have the enzymes to do both. Therefore, these are going to be relatively complicated organisms, more complex because they have the enzymes for aerobic metabolism as well as the enzymes for anaerobic metabolism. Okay. These are survivors. Okay. The anaerobes going to be at the bottom of the tube. Okay. Oh, and if I didn't mention, I'm sorry. The facultative anaerobes, you're going to see them throughout the tube because they can survive anywhere. Up in the oxygen, without the oxygen, they see lots of versatility within the tube. Okay. The anaerobes, we're going to find them at the bottom. They're going to get away from the oxygen because remember, they don't have the ability to detoxify it. Too much oxygen is going to poison these organisms. Okay. And then the aerotolerant anaerobes, surviving in oxygen, they can detoxify it, but they're not going to utilize it. Just like this facultative anaerobes, doesn't matter where they grow. Okay. They're going to do well at the bottom of the tube, at the top of the tube. Okay. So they can break it down, they can detoxify it, they're just never actually going to utilize it. Okay. Now, the capnophiles are moving away from the oxygen. Capnophiles grow best at higher carbon dioxide levels. Okay. Usually they like levels that are actually much higher than atmospheric CO2 levels. If you want to culture these in the lab, you actually have to put them in an incubator and pump carbon dioxide in to keep them stable. Okay. Examples of organisms that are considered capnophiles, Neisseria, things like gonorrhea, meningitis, brucella, streptococcus pneumoniae. These organisms prefer, okay, the high CO2 environment. An example of this and a use of this, we don't really see this much anymore, but utilizing what we call candle jars, and it is literally a candle in a jar, to create a high CO2 environment. Because if you put your bacteria into the jar, light a candle in the jar, and then screw the lid closed, eventually the candle will basically use all the oxygen within the atmosphere of the jar. Okay. And will release carbon dioxide, so you can actually create a high carbon dioxide environment within the candle jar. Now, we don't use these obviously anymore in any type of clinical situation. This is what's known as a gas pack, and gas packs basically will do the same thing. You see the little pressure, okay, spout here. You can hook a hose to these and actually suck out the excess oxygen and or put in the carbon dioxide concentration that you prefer. So let's talk about how microorganisms are processing oxygen. As oxygen enters cellular reactions, it gets transformed into several toxic byproducts. So as oxygen is utilized, these are the results of that oxygen utilization. Singlet oxygen, okay? Extremely reactive molecule that can damage and destroy a cell by oxidizing membrane lipids. Basically what we see happen is that free radicals oxidize membrane lipids by pretty much tearing away, okay, important molecules, removing things like electrons. So free radicals attack membranes. That is going to be bad. So cells are not going to want to leave these singlet oxygen molecules roaming around inside of the cell. Okay? That's going to be an issue. So there's got to be a way to get rid of these, and we'll talk about a few examples of that in just a minute. Okay. Superoxide ions, also highly reactive free radicals. Okay. Hydrogen peroxide is an example of this, a very common byproduct of oxygen metabolism. It's actually toxic to cells. We commonly use it as a disinfectant and an antiseptic, getting rid of microorganisms that are unwanted on tissues or on surfaces. Hydroxyl radicals, again, also highly reactive. Most of these work by being toxins or by destroying nearby molecules, and that's very, very bad for the cell. Okay. So let's talk about some of the more commonly made ones. Okay. So if you are going to be an aerobic organism, you have got to be able to detoxify oxygen. You've got to be able to get rid of those highly reactive oxygen species that we were just talking about. Okay. So most cells develop enzymatic pathways that will find and get rid of those reactive oxygen byproducts, not necessarily by destroying them, but usually by turning them into something else that's not going to be reactive. Okay. The most common process is the one I'm going to show you. It's a two-step process, okay, where that superoxide free radical, we see it here, okay, is complex with two free hydrogen ions, okay, with the help of an enzyme called superoxide dismutase, and that superoxide ion actually is converted over to hydrogen peroxide. Okay. Well, that would be great, okay, except what? Hopefully in your head you're thinking, wasn't hydrogen peroxide on the list? Okay. Yes, it was. Hydrogen peroxide was absolutely on the list. We turned one problem into another problem. So this is going to be a two-step process because we've also got to get rid of the hydrogen peroxide. Okay? Hydrogen peroxide is converted into water and oxygen with the enzyme catalase. Okay? So catalase catalyzes the conversion of hydrogen peroxide into water and oxygen. Okay? And this is doublet oxygen. We made some here and we made some here. Okay? That doublet oxygen is not reactive. Okay? Doublet oxygen is pretty stable. Okay? And it can be utilized elsewhere within the cell. And we all know that water is also going to be non-reactive and stable. So we turn these reactive oxygen species into non-reactive products eventually. Okay. So having enzymes like superoxide dismutase and catalase give anaerobes the ability to live in oxygen-rich environments. Okay. Chances are, if you are an anaerobic organism, you would not have either of these. So another factor, pH. Okay. Bacteria grow in a variety of pH values. We see what we term acidophiles. This is a nice one because the word and what it does is right in there, phile to like, acido, acid. Acidophiles live in mildly or acidic pHs. The example I have here is lactobacillus. It tolerates a mild acidity. It's going to be happy as somewhere around that 5.4 range. Less happy around the 2 or 1 pH. Okay? But there are absolutely bacteria out there that live in extremely acidic environments. Okay? Euglena mutabilis, Euglena mutabilis grows in acid pools somewhere between pHs of 0 and 1. Okay? We see thermoplasma living in coal piles at pHs of 1 or 2. These are definitely extreme versions of acidophiles. So again, this is test-like questions. Expect examples of organisms and to be able to tell me, because they're living at those low pHs, we would call them blank, and hopefully you could answer acidophiles. Okay. Picrophilus thrives at pHs of about 7, but could live at a pH of about 0. So wait a minute. Let's think about this one. Okay. Couldn't we also call Picrophilus something else? Okay. What does it mean when an organism can survive? Look, it's right here in the PowerPoint. Can survive at these low pHs. Couldn't we just as easily call this a facultative acidophile? Okay. It could live at those low pHs if it had to, but it's perfectly happy at a neutral pH. Speaking of which, Okay. Neutrophiles. Okay. Happy to have those neutral pHs somewhere between that 5.4. That seems to be the threshold for a neutrophile, all the way up to a pH of about 8. Most organisms, preferentially somewhere around that pH of 7, they like it relatively neutral. Okay. The alkalophiles prefer alkaline pHs. No, they're not called basophiles. They like the higher pHs. Okay. Natromonas live in hot pools and soils at pHs of about 12. So these will be alkalophiles. And because they live in hot pools, what else could you call them? Hopefully you're thinking thermophiles. Okay. Proteists can actually create their own alkaline conditions to neutralize things like urine and to help colonize and infect the urinary system. So these guys will actually exude alkaline substances to actually neutralize acidic pHs. Okay. Most bacteria are going to be preferentially neutrophiles. Okay. Now, when it comes to bacteria that live within our bodies, it depends on where they live. Okay. If it's an organism that thrives in the stomach, chances are it's an acidophile. If it's an organism that is particularly happy in the liver, well, your liver is about pH 9, so it's probably going to be an alkalophile. But pretty much the rest of your body, okay, with those few exceptions, are going to be neutrophiles. Okay. Another example of acidophiles, the vagina actually has a relatively acidic pH. Another environmental factor, osmotic pressure. Okay. Osmophiles live in habitats with high solute concentration. So this is a general term for organisms that like high solutes. So if you remember a solute, anything dissolved within the environment, anything dissolved within the liquid. Okay. These can be sugars, salts, metals, you name it. Okay. Usually sugars and salts kind of take the lead here. Halophiles are a specific type of osmophile. Okay? We've dealt with them on the MSA plates in the lab. They prefer high concentrations of salt. Okay? So, if it likes salt and sugar, you can't call it a halophile. Okay? Halophiles are specific to salt-loving organisms. If it likes salt and sugar, it doesn't care what the solute is, you would term it an osmophile. Okay? Now, those terms that we were throwing on to the beginnings of some of these words are actually quite useful here. We see obligate halophiles like Halobacterium and Halococcus that grow in solutions of 25% salt, but their requirement is at least 9% salt. That is an extremely salty environment. Okay? Those organisms, because they require high salt concentrations, are considered obligate halophiles. Obligate like an obligation, meaning that you have to do it. Okay. Facultative halophiles can survive in the salts, even though they don't normally reside in particularly salty environments. So here's two of those predecessor terms thrown in, obligate and facultative. Okay. Staphylococcus aureus is the one that we dealt with. That was our halophile example in the lab. Okay. Staphylococcus aureus can grow in salt medias ranging from anywhere from 0.1% all the way up to 20% salt. Okay. So it's got a wide range of halophilic capability. Okay. The barophiles, particularly interesting, are adapted to high pressure environments, specifically pressure that is atmospheric pressure. Usually these are deep sea microorganisms. They are strictly adapted to high pressures, so much so that they're so used to pushing back against those high pressure environments that if they're brought up to normal atmospheric pressure, they usually rupture from the inside. Okay? Mainly because these organisms, in order to stay structurally sound, are having to adapt themselves to push back against the atmospheric pressure. Okay. Now, Another environmental factor that we see and that we can't ignore is the fact that organisms are going to live with other organisms. Okay? In all but the rarest instances, microorganisms live in shared habitats. Okay? They're going to have to deal with other organisms being within their environment. And we see these associations between organisms in two varieties, what we call symbiotic relationships and non-symbiotic relationships. And even these actually have their own subcategories. Okay? In symbiotic relationships, there's a requirement. It's usually a nutritional relationship. We're sort of feeding one another or working together. In a mutualistic relationship, both members of the relationship benefit. Okay. I like to use us and E. coli as a great example because we give the E. coli nutrients and a place to live, and the E. coli actually helps us digest some nutrients, and by breaking them down provides us with vitamins that we actually need. Okay? We both benefit from the relationship. It's mutualistic. Okay? Now, in a commensal relationship, one member of the relationship benefits, but the other is neither harmed nor does it benefit. Right? It's just kind of a, hey, you happen to be their relationship. So an organism that would feed off of excess, right, would be an example of a commensal organism. These are actually a little harder to find examples of. Most of the time, relationships end up being somewhat mutual if they are not going to be parasitic. These are the two most common. Okay? Now, in a parasitic relationship, one member benefits, that would be the parasite, while the other member is harmed, that would be the host. Okay? So the parasite benefits from the relationship and the host is harmed. Now, in the non-symbiotic relationships, the relationship isn't required. These are just organisms that happen to be living in the same space or happen to be living together. Okay? They don't have to live together, but they do. Okay? In a synergistic relationship, members cooperate. Everyone's working together. Think about certain types of bacteria that are living in biofilms. Do they have to live in the biofilms together? No. But if they live in the biofilm together and have this nice communal relationship, and both members of the relationship benefit from the relationship, they're having a synergistic membership. Okay? Now, in an antagonistic relationship, and we've mentioned microbial antagonism before, some members get inhibited or destroyed by others. Okay? Basically, some members outdo or outcompete, or in some instances just attack, okay, the other member of the relationship. This was our bacteriocins. I told you it would come up again. Okay? If you remember, I said this was like bacterial mace, where the bacteria attempt to invade and push bacteria out of their normal habitats within the body, and the bacteria that are being attacked actually end up secreting chemicals that destroy their attackers. This is an antagonistic relationship. Again, the best example of synergy, I like this particular title, the epitome of synergy, are biofilms. Okay? These are mixed communities of bacteria. They do not have to be living with one another. Okay? But because they are living with one another, members of the biofilm actually benefit. Okay? So they end up being this multi-layer conglomerate of cells. One of the most interesting things about biofilms, okay, is that biofilms communicate. The bacteria within the biofilm communicate with one another, and it's not like picking up a phone. It's chemical communication through a process called quorum sensing. Okay? And in quorum sensing, bacteria actually end up producing molecules called autoinducers. Okay? And the autoinducers serve as sort of warnings or calls to other nearby bacteria. As the population density of the organisms change, the autoinducers begin to be slightly different. Basically, some of these organisms are going to send out chemical signals saying, hey, get ready. We've got an antibiotic coming our way. There's an attack coming. It's a sort of an early warning system, but for good and bad reasons, including things like food. Okay, there are extra nutrients out here. Be prepared to take it up. Okay, there are phagocytes and inflammation and white blood cells policing this area. Find a way to protect yourself. Okay, so quorum sensing, we're sending out chemicals that basically tell nearby organisms what to expect. So I'm going to do this last little bit talking about bacterial growth. One thing I want to definitely mention is that growth has nothing to do with size when it comes to bacteria. Do not write down or fill in the blank with anything like size. Okay? Microbial growth is an increase in the number of cells. It's amount or population. Your book likes the term population. Okay? Now, bacteria reproduce through a process called binary fission. This is the simplest of cell divisions. It actually tells you what's going to happen in the word. Binary means two, fission means to divide. These guys are going to divide in two. Okay? Now, what we see is that a cell begins to prepare itself to divide. One of the first things it does is reproduce genetic material. Okay. Once the chromosome is replicated, interestingly enough, a part that gets kind of skipped in here is that these chromosomes end up attaching, okay, to the cell membrane of the cell. And then as the cell gets larger, as the cell itself grows larger, that is what's going to pull the chromosomes away from one another. Okay? There aren't going to be any spindle apparatus or metaphase where the chromosomes line up in the middle. They get separated because the cell gets bigger. Okay? Now, once the chromosomes are actually moved to opposite sides and separated from one another, a protein band forms down the center of the cell. Think about it like a rubber band. Okay? That over time shortens, okay, and begins to pinch in, it's basically pulling in actually, the sides of the cell. So these sides get pulled in, okay, and once the septum, okay, is complete, so once we've pulled in from both sides equally and sort of moved that membrane in together, okay, the cells are considered divided. This would be the result if your cells were going to be things like individual bacillus or individual coccus. Cells that do not completely separate from one another after division are what cause arrangements. Okay? So if they stay attached and form chains or clusters, then those are how the arrangements of cells arise. Okay? So this is binary fission, a very, very simple version of cell division. You'll notice that at the end of it, we went from one cell to two. So we will always see cells double. Okay? So in the next lecture, I'm actually going to pick up with binary fission again. So we'll do a review of binary fission, and then I'm going to talk a little bit more about cell growth, measuring cell growth, and doubling times and generation time.